A good afternoon to Dr. Suresh and to whoever that is watching this video. I hope that you find this video useful and very knowledgeable. So I will be presenting on protein domain prediction. This is our assignment 1 for bioinformatics. This is from our chapter 2 sequence-based domain databases. So my name is Yu Shun. I chose this topic is because I find that this topic fully applies the 3 concepts of bioinformatics, computer science, molecular biology, and mathematics. So, without wasting any time, I will be explaining about our core concept that I will be presenting on this video. So first of all is the protein domain, the protein domain databases, the protein domain identification methods, which are the tools, the software, and most importantly, the mathematics behind the concept. Based on my research, until now, protein domains do not have a proper definition. They are just defined as a structural, functional, and evolutionary units of protein. So the domains can work independently or cooperate with other domains to have a different functions for the physiology of the body, for the homeostasis, and such. So for now, we will be entering the protein domain databases. So there are a lot of variety of the databases, but I chose one that is the most comprehensive domain family databases, which is PFAM. It is manually created using a seed alignment first. So as you can see on the image there's a seed and extend strategy. So this is the method used for the alignment. So and then they would use a profile hidden Markov model, which is built based on the seed alignment. Okay, so the profile hidden Markov models are probabilistic models that encapsulate the evolutionary changes that have occurred in a set of related sequences. So after the profile is created, it is queried against a sequence database called a PFAM sequence, or just PFAM SEQ. So this is actually derived from UniProt KB. So PFAM and UniProt are all from EBL. So of course they will be related. Not only that, the profile HMMs are actually used for full alignment to improve the probability in the model, which may lead to a slightly different alignment. So of course, the more it's just like mathematics. The more data you have, the better it is. The more accurate your output will be. So how do we enter PFAM? We can just actually Google PFAM, and then we can just enter the site through Google. So when you enter the site, you can put in the sequences that you want, or you chose from UniProt other knowledge base, which is from EBI, EMBL. So for this example, I chose is the P element induced wimpy testis for the fruit flies. So this is a regulatory protein. After you press search button on the PFAM below the sequence box, it will query for the result. So for PFAM, you get actually a variety of results, the chemical properties of the protein and the structure of the proteins. So for this, you can actually pinpoint the accurate function. So this PV protein is a regulatory protein, as I said just now. It's responsible for stem cell and germ cell differentiation. It is being studied on human for the stem cell therapy. Not only that, you can actually find the domain organization for the PV protein. As you can see below, there's a green, red, blue, purple, yellow, and the orange, which is the PV. So you can actually find the whole set of the family there. So based on the statistical research that I found about UniProt knowledge base, which is the database, as shown in the graph 1, there is an increase of the UniProt KB and its coverage for PFAM, meaning since 2015 until 2020, there's an increase in the coverage for PFAM, and then there are around 180 numbers of sequences in the millions, 180 millions. That's a lot of sequences in the database. So on the right side, you can see table 1. So table 1 actually shows the type of PFAM actually. So you got family, you got domain, and so on. So of course, the software that would need a patch. So a patch is basically like an update of the data or a new features. So based on the table here, there are around 10,000 new families, 6,000 plus of domains, 200 plus, around 300 of repeats, and so on based on the table. So these are all the new proteins that have been uploaded into the database. Now we move on to the protein domain identification, meaning we will have to use some tools, right? So the tools that I chosen is called SMART. It's a simple modular architect research tool. It's actually also by EMBL, as you can see on the left side here, which has 2 modes, which is the normal mode and the genetic mode. So this most of these softwares tools, they use hidden Markov models. The concept of it, which later I will explain on the on the part of the mathematics behind the software. So as you can see on the right side there is the home page for the smart genetic mode. You can query in the sequence that you get from UniProt or assemble. So once you press the sequence smart, you will get the domain as the first bar that you see on the top. As you can see, it's a gray, which stated there PFAM PK. So actually what I queried was pyruvate kinase, which is a protein that is used in phosphorylation during glycolysis. So you can have all the details. You just have to press the button and then you can get all the details, the domain, and so on. As you can see below the slide, you can actually see an homologous of non-structures. So these homologous are actually important to be multiple aligned, because this is used to construct the profile hidden Markov models. So after you have all the information that you have gotten from the database, which are very accurate, you can actually write it down, copy paste it down, just like what I did. For my case, I chose pyruvate kinase. So the function is that it catalyzes the final step in glycolysis, which is the phosphorylation of ADP to ATP. And then the end product is also pyruvate. So you would see that it belongs to a PK beta barrel domain like superfamily, and another pyruvate kinase beta barrel domain family. So as you can see on the right side there's like a 3D moving 3D model. This is actually for the human pyruvate kinase. So this image I got it from JMOL, which you can get the code from PDB and put inside JMOL. So there are four domains: a small N-terminal helical domain, a beta alpha barrel domain, a beta barrel domain, and a 3-layer alpha beta alpha sandwich domain. So these are the long name actually, but if you did more research, it's actually just domain A, B, and C, but there's a really long name for that. Then not only that, you can get the molecular function of the protein that you queried in this smart tool. So one of it is magnesium ion binding, which magnesium ion is a cofactor, and pyruvate kinase activity, which is regulating the cell metabolism. Potassium ion binding controls the kinetic mechanism, and ATP binding, which is important for coenzyme and enzyme regulation. So now I will get into the part where people don't like the most, which is the mathematics part. For me, I like mathematics because without mathematics, nothing can advance. Technology, whatever it is, software, nothing can work. without mathematics. So the mathematics behind protein domain prediction is actually, first of all, is dynamic programming, that is for sure. So it is the combination of both mathematical optimization method and computer programming method. So meaning you use the logic of mathematics for programming, which actually are the same thing. Without the logic, there's nothing at all. And then second is the hidden Markov models, which we heard a lot. If we mention about protein, that's the first thing that we would think of. So it is a statistical Markov model in which the system is being modeled, is assumed to be a Markov process. So as you can see on this slide, dynamic programming is a useful mathematical technique for making a sequence of interrelated decisions. It provides a systematic procedure for determining the optimization, the optimal combination for decision making. So a lot of people do not know that actually global alignment and local alignment are both dynamic programming. This is why I mentioned that mathematics are very important, is because dynamic programming needs mathematics. Most of the thing that we use now, everything is mathematics. So as you can see on the right there, there's a small image, a small table actually. So this is a Blossom metric system, as I'm sure anyone taking bioinformatics would know. So actually, how does this work, So first of all, it's the graph theory, okay? Graph theory is the study of graph, mathematical structures used to model pairwise relation between objects from a certain collection. But in bioinformatics, a graph is a collection of vertices or nodes and a collection of edges. It is not what we think like 1 plus 1 is 2. No. In bioinformatics, mathematics, graph, it is different. And then the second would be matrix theory. Just like I said, the Blossom matrix. Matrix is described in the field of matrix theory as being added, multiplied, decomposed in various ways. So it is a sub-branch of linear equation at first, a linear algebra at first. Matrix algebra has now grown to cover subjects related to graph theory, combinatorial, and statistics. Later, I will show you the images of both theories and how it is used in bioinformatics. Okay, now you can see there are around a few pictures. Actually, there are three pictures. First is protein-protein interaction, also known as PPI. This uses graph theory. And then as you can see on the right there, is a gene regulatory network. This is also used from graph theory. And then the last one will be the metabolic networks, as you can see at the bottom left. All of this uses the graph theory. So in this part, the mathematics is not what you think it is. It is finding the shortest way to solve a problem. In programming or computer science, this problem is known as a traveling salesman problem, which is the shortest route to deliver all the sales, all the products. I have to remind that as you can see on the protein-protein interaction on the image, the green circle is known as the node, while the line are known as the arrow or the vertices, which links from one point to another. So now we move on to the hidden Markov model. So like I said just now, it is a statistical model in which the system being modeled is assumed to be a Markov process with unknown parameters. And the challenge is to determine the hidden parameters from the observable parameters. Meaning, you determine the future based on what you see. Because the outcome you cannot determine, so you have to use what you can see to determine the outcome. So as you can see on the top right, there's an image showing hidden and observed. So the hidden is the weather. We cannot determine the weather. We are not God or anything. But we can determine the clothing of what we wear. If it is a hot day, we would wear something short. A mild day, then you would wear something like I did. For a cold day, you would be expected to wear something cold. So this is one of the simple HMM question. And then to calculate the probability, you would have to use Bayes' theorem, which is calculating the joint probability, meaning the probability changes based on what has happened, what you had seen. So just like the image on the bottom left, you would see a very confusing graph. You can use a graph theory to solve this. Actually, it is very simple. You can just think of x1, x2, and x3 as three boxes. y1, y2, y3, and y4 are the cards inside the boxes. So there is a very long permutation of probability that you can make from. So now we will get to profile HMMs. So it have a formal probabilistic basis and have a consistent theory behind the gap and the insertion score. In contrast to the standard profile methods, which is the heuristic methods, HMMs apply a statistical method to estimate the true frequency of a residue at a given position in the alignment from its observed frequency, whereas the standard profile uses the observed frequency itself to assign the score for that residue. So as you can see, the image above, so it starts with a multiple sequence alignment, and then the insertion deletion can be modeled, and then the occupancy and amino acid frequency at each position are encoded, and then the profile is created. So this is just the front part. This is just the tip of the iceberg for the profile HMM, because the hard part is the programming. You will have to use mathematical methods and your computer science methods and programming methods. So for bioinformatics, especially in protein domain analysis, which is this chapter, we would have to be actually to be strong at all the concept of mathematics, programming, and biology. So for the conclusion, I would say that the protein domains are the basic unit of protein that can fall, function, and evolve independently. So meaning that we cannot control proteins. That's why the knowledge of proteins domain is very critical for the classification, understanding their function, and annotating their mechanism, or just designing a protein. So I would say that there are a lot of methods, databases nowadays because of the advancement in technology. We would just generalize as sequence based and structure based. So for my presentation, I'm using all the methods, all the database, all the tools are from sequence based analysis. Because based on this assignment, we are required to be based on the sequence based analysis. So I will not show the structure based for this presentation. So thank you very much for watching.